I am going to introduce our first speaker this evening is Bill Hack, and he is the founder and CEO of Cadex Genomics, excuse me.
Greetings. Cadex Genomics is a molecular diagnostics company that has developed a blood-based tumor burden platform which runs on PCR and has multiple applications in the field of oncology. Hi, my name is Bill Hack. I am the CEO and one of the co-founders of the company. The problem we address with our first product is to improve on the current standard of care for assessing the effectiveness of therapies delivered to cancer patients with stage 4 solid tumors. Currently, imaging is used to assess whether a patient's therapy is working. Imaging is typically performed in 8 to 12-week intervals. The problem with this approach is that when the therapy is not working, the patient's tumor is left to grow unchecked for weeks. Additionally, the patient is receiving all the toxic side effects of those ineffective treatments and none of the intended benefits. And finally, it is a tremendous waste of healthcare resources to deliver multiple cycles of ineffective treatment. Many of these treatments cost tens of thousands of dollars, and patients pay an increasingly higher portion of those costs. Our solution to this problem is Alebrix. Alebrix is a real-time, blood-based therapy monitoring tool. We compress the time it takes to evaluate the effectiveness of a patient's treatment from 12 weeks to just 12 days. And most importantly, after just a single cycle of treatment. Alebrix has the potential to improve patient health outcomes while significantly reducing the cost of cancer care. This is a $3.5 billion market opportunity. The way our technology works is conceptually very simple. When cancer cells die, their DNA is shed into the blood. This is called CFDNA, or cell-free DNA. All other things constant, the larger a tumor, the higher the concentration level of CFDNA in the blood. So by comparing the CFDNA concentration level in the patient's blood prior to treatment with the CFDNA level 12 days after treatment, we can determine if a patient's tumor is growing. The reason our test is so accurate and precise is we target a section of the DNA called retrotransposable elements. This part of the DNA has 1,800 copies in every cell, giving the assay unmatched analytic sensitivity. To make the assay cancer specific, we filter out non-cancer CFDNA by exploiting the fact that cancer cell fragments, DNA fragments, are shorter than non-cancer CFDNA fragments. Our approach has many advantages over approaches of other CFDNA companies. These are, we require no changes in the patient workflow. Our technology is extremely cost effective. The cost of goods for analyzing both blood draws is under $50. And we are the only company with data demonstrating we can monitor therapy effectiveness in real time. We have studied our technology in over 175 patients across 3 studies. The first of these studies is a proof of concept study comparing healthy patients against treated and untreated stage 4 colorectal cancer patients. The results demonstrate that our technology can distinguish between these 3 cohorts with an area under the curve of 0.989. The second study, which we conducted with MD Anderson, determined that within 2 to 3 weeks, Alebrix can identify 63% of patients with progressive disease at 8 to 12 weeks using imaging. Most important, there were no false positives, 100% specificity. And in our most recent study, we were able to identify 80% of patients who were classified as having clinically relevant progressive disease using imaging at 8 to 12 weeks. Again, with no false positives, 100% specificity. Here are the key company milestones. Looking forward, by the end of this year, a Librix will be finalized and will be ported to a point-of-care PCR platform that will give us tremendous scalability and the ability to easily move to markets outside the US. The developer of this point-of-care platform is a strategic partner for us who is funding large portions of our R&D. They have already expressed an interest in acquiring the company. By the end of this year, our first clinical validation study will be completely enrolled. The data from this study will be evaluated by the end of Q1 of next year, at which point we will commercially launch a Librix for stage 4 colorectal cancer. By the end of next year, we will expand the Librix into lung and breast cancer. We will also receive Medicare coverage by the end of next year, which we believe is the optimum point to exit. Here's the team you will be investing in. All 6 of us has 20 plus years experience in life sciences, bringing novel drugs and diagnostics to market for cancer patients. We have worked at companies like Genentech, Veracyte, Invitae, Castle Biosciences, and others. This experience is in fields such as R&D, clinical operations, medical, sales, marketing, and reimbursement. I thank you for watching, and I look forward to hearing from you soon. Thank you.
Bill, thank you so much for your presentation tonight. You know, this seems like a really, really exciting technology. And as you mentioned, the idea that patients really would know a lot faster whether their treatment is working, patients and providers, I should say, as well as the fact that they, you know, hopefully would suffer less side effects. We all know that cancer treatment is not a fun process to go through. So very exciting. I'm going to start off with a couple questions, and then I know those in our audience are putting some questions into our Q&A function. Again, that is just to the right of the chat. I would love to take your questions here in just a minute, but I'm going to start off with a few of my own. So you mentioned that colorectal cancer, breast cancer, and lung cancers are kind of first in your kind of pathway. Can the technology be used for all types of cancer?
We believe so. The you know, it's generic. The marker that we're looking at is fairly generic to cancer, meaning we're looking at these retrotransposable elements, non-coding regions of the DNA. And so those are shed by every tumor type. And so we think the assay will work across all tumor types. But the proof is in the pudding, and we need to do further studies to prove that that is the case. So for now, we do have evidence for lung, breast, and colon. And we'll need to do additional studies to actually demonstrate that it works across all tumor types.
Wonderful. And you talked a little bit about the technology itself. And my understanding is you're looking at kind of the length of the CFDNA markers to determine if this is tumor or non-tumor CFDNA. Can you talk a little bit more about that?
Of course, and that would be a great question, by the way, to re-ask when we break into the group the table
networking.
Yeah, the networking. Our chief scientist will be there, and he can answer this with far better precision than I can. But that is correct. So there's really 3 things that that really ensure that we're looking at CFDNA from tumor types, from the tumor as opposed to non-cancer cells. And the first thing we do is we can sort of cancel out the
the background noises, if you will, because we do take a blood draw before therapy. And if there is background noise from non-cancer DNA, and those cancel out because we compare with, with a blood draw after therapy is given. And so that background noise cancels out. The other thing we do is in our clinical study, we don't study patients who really, and most of these patients aren't really eligible for chemotherapy anyway, but patients with conditions where there's high levels of necrosis and apoptosis from non-cancer sources, such as very severe inflammation and patients with infections. But the third thing is that we're really excited about is that we can filter out CFDNA from those sources by looking at that fragment length. As it turns out, cancer really sheds DNA that is fragmented into these much shorter fragments. And so by looking at those short fragments, we're able to really filter out non-cancer sources of CFDNA. And in our studies, we actually spiked, if you will, using white blood cells, the CFDNA levels to the point where the amount of CFDNA went up fivefold. And when we filter out those long fragments, the CFDNA level is much lower, and exactly what you would expect from a patient who is not progressing. So we really have good evidence to support that these three methods really do get down to cancer-specific CFDNA.
Well, and we have some great questions from our audience. Again, I'm hopeful that those of you asking these questions will join us in the networking, because these are getting pretty technical, but I'm going to throw them out there anyway. I know. So, do what you can. And if you want to say, we're going to delay until networking, you can do that. Interesting concept. Definitely something which is needed in the field. The question here is, there have been some studies that show that the correlation of CFDNA with radiological progression is still an imperfect science. They put a study in here highlighting that evidence. Can you speak to that a little bit?
Sure. So, I mean, our studies certainly show that we can do this with a high level of specificity. And specificity is really the most important metric for this field. So, the idea here is that physicians have to have confidence that when we identify with a Librix a patient who is progressing, that in fact that patient is progressing and their therapy is not effective. And that's the most important metric. And so what we've done here is in all of our studies, and going forward, we will optimize the assay for specificity. And we have been able to identify so far 100% specificity. And the methods that we use are very different from other CFDNA companies, which is why we've been able to achieve a 100% specificity. If you want, additional information on it, I really do encourage people to come to our networking session, and our medical chief medical officer and chief scientific officer can answer the question probably in more detail than I can.
No, I thought that was great. And your data are certainly very impressive. So another question, and I'm just going to read a little bit, but the problem with stage 4 cancers is that most of the time the treatment options may be limited, and that switching therapy too early might actually disable patients to receive benefits from therapies which may take longer to shrink tumors. Can you address that concern?
Sure. So the physicians we've spoken with and the evidence that we've seen, especially from our MD Anderson study, where we took serial tests and we were able to see how a patient was progressing and how we were able to identify patients whose CFDNA levels were going up or down. And in no case there did we see a patient who was progressing, and then in further on down the line, their disease started to they started to respond to therapy. So if a patient's going to respond to therapy, it's going to happen from first therapy on. There are certainly some studies initially with immunotherapies where it appeared as though patients did not respond initially and then later on started to respond. But further studies have suggested that these are really patients who are exhibiting what is known as pseudo-progression. And in fact, they weren't really progressing. It was simply a facsimile of imaging that was picking up inflammation around the tumor. And again, I think Dr. Knopf can address that question better than I certainly can.
Awesome. Well, I'm going to encourage everyone in our audience to come to networking. And I certainly will be at your table to hear more answers to these questions. I would love to hear from you. It seems like this assay would also be able to answer questions like, could the patient take a drug holiday or a treatment holiday? How long do they need to be on the treatment? And again, I realize these are probably things that would need to be studied more, but can you address that a little bit?
No, we certainly agree that really any sort of de-escalation of treatment, our assay will be able to help address those questions. Like, for example, take a drug holiday, observe them with this test to see if they're progressing and progressing in an aggressive way, and get them back on treatment. Also, potentially try to take out one of the therapies in their in the bucket of therapies that they're on, and take one out to see what's going on. So there is opportunity to experiment with treatment within obviously reason, and observe what's going on in a real-time manner. So I quite agree with you that there are some very interesting opportunities for therapy de-escalation.
All right, so one last question for you before we move on to our next presenter. Who are your competitors for this type of monitoring? And what are your advantages over your competitors?
Sure. So probably our biggest competitors, potential competitors, are Guardant Health and Natera, two very large CFDNA, ctDNA competitors. And I mean, right away, the biggest advantage is data. To my knowledge, one is they're keeping the data that they've generated very close to the vest, and I have no idea. And I've asked physicians who are close to their company, what are the performance characteristics of their test in the therapy monitoring area, and no one seems to know. So the other thing is the Natera, which does have a publication, their treatment was, or their second blood draw was taken after the second cycle of treatment, and so they really have no data supporting the use of their test in a in a real-time basis, that is after a single cycle of therapy. And then other advantages, much less expensive. Those tests cost in the order of $3,000-$4,000. Their cost of goods is above our intended list price. And then finally, because of the nature of their testing, where they're looking at ctDNA, they have these very, very wide band, if you will, assays that look at many different really dozens of different mutations. And invariably, they have patients who they'll run the test on who they can't yield a result because the particular mutation for that patient is not in their panel. And in fact, the Natera data, they were only able to really yield a result on 75% of patients whom they tested. So
wow,
Yeah. So
Yeah.
yeah, and we were able to generate a result on every patient so far that we've been able to that we've analyzed in our studies. So those are
Fantastic. Those
Thank
are all great things. Again, thank you for being here tonight, Bill.